My name's Erin Alberts. I'm going to be talking about the Tor complex, specifically a part of it called Raptor. I think that's about it. Today is November 12th, 2025. So, this is Raptor, otherwise known as the regulatory associated protein of Tor. This is the TORC1 complex. This is Tor. So it's made up of 3 genes, which is confusing. But this is Tor. The colors are not very visible, is what I'm realizing. It's the pink part. Raptor is this blue part, and LST8 is the yellow. It's green on here. It's the yellow part. TORC1, or the Tor complex, is essential for growth and development in all eukaryotes. That includes plants, mammals, amoebas, etc. Each piece of the Tor complex has its own part to play in how Tor functions. Tor is the active site. It's a kinase, didn't mention that. Tor contains the active site. Raptor recruits substrates to Tor, and then LST8 maybe helps as a chaperone. It does some things. Not 100% clear, but it does things. There are 2 raptor homologs in Arabidopsis. I'm going to be talking about Arabidopsis, but also Nicotiana benthamiana, and I will make sure to be clear when I'm talking about one or the other, because a lot of stuff is clear in Arabidopsis, but not so much in Nicotiana because it's not as widely used. So in Arabidopsis, there are 2 raptor homologs, raptor 1A, which has low expression, and raptor 1B, which has high expression. I do have a thing. This is just in the various cell types in the plant. You can see that Tor pretty much always has high expression. Raptor 1, so this is raptor 1B. Raptor 2 is raptor 1A. Raptor 1 has pretty high expression throughout the various cell types in the plant. Raptor 2 or raptor 1A has pretty low expression. I think that's all I want to say about that. One of the reasons that we have chosen to study Raptor is that Tor mutation, so the Tor protein, this one, is lethal in Arabidopsis. It's also lethal in many other organisms. So these are normal, these are Arabidopsis embryos. These are normal embryos. They're growing. These are Tor mutant embryos. Basically, they just don't grow. They're dead. And that's important because Raptor 1B mutation has been shown to be embryo lethal, but also to not be embryo lethal. And so the question sort of is, well, I guess I'll get to that in a second. But so in this paper from 2005, by not the Anderson that I worked with, they showed that when you get rid of Raptor 1B, you get these teeny tiny little plants, but they're still alive. This group in the same year showed that when you mutate Raptor 1B, the seeds are not alive, they are not viable, and they just don't grow. That's the tiny little seed, that's a normal seed. So that's one discrepancy. And then as the years go on, you can see that, so these are all Raptor 1B mutants. You can see that they get bigger and bigger, sort of as the years go on. Not actually the plants, but what people are showing in their papers is a plant that is more and more alive. So then by like 2018, you have plants that are basically like half the size of a normal plant, as opposed to what this paper showed, which was that they are dead, or this paper, which shows that they're really tiny. So basically, we're asking the question, is Raptor required for development, and can the Tor complex function without it? Specifically in plants, but this is also has some applications to things like cancer research, medical, like pharmaceutical targets. Is Tor able to function without Raptor? Because there are things that target Raptor that are used to treat certain medical conditions. So there's three ways that I'm going to, or am in the process of, assaying whether or not Raptor is required for development, growth, function of a plant. The first one is using CRISPR-Cas9 to knock out the Raptor genes in Arabidopsis thaliana. Like I said before, there are two Raptor genes, and some people have a theory that when you knock out Raptor 1b, potentially it's being compensated for by Raptor 1a. So that's something that I will definitely be testing. The second is virus-induced gene silencing of these genes in Nicotiana benthamiana, so in a second model system. And then I'm also going to be doing, or am doing, genotyping and selection of Salk and Sale lines, which I'll talk about at the very end. So I have been working with this system called CRISPR Combo for like the last 3 years. And basically what it allows us to do, I'll go through the protocol on the next slide, but what it allows us to do is generate potentially a homozygous knockout mutant for Raptor 1b and Raptor 1a. I'm starting with Raptor 1b because it's the more highly expressed. It's the one that we have the most data on, but eventually both of them will be tested. So I've designed these guide RNAs to cut at both ends of the Raptor 1b gene so that it's completely gone. So you target here, and you target here, and then it's gone, so that we can be sure that there is no transcript of Raptor 1b being made, that there is no way that there's any protein that could be performing the same function with this gene. So this was a pretty cool protocol. It came out, in 2023, so it's only been 2 years, that uses this very special or innovative way of generating double-stranded breaks. You can also use it to overactivate, though, which is part of the interesting part about it. So you have this, your regular Cas nuclease, and you have these SUNTAG activators. Basically what happens is that you design your guide RNAs to be a certain length, either 15 nucleotides long or 20 nucleotides long. And depending on the length of your guide RNAs, the Cas nuclease, not the Cas nuclease, the guide RNA scaffold, either directs the nuclease to cut at where your guide RNA is targeting, or it recruits these SUNTAG activators, like so many of them, to the site that you want to modify. So the way that they have utilized this special Cas9 system is that you can make a guide RNA that targets flowering locus T specifically, and then you can combine it with one that targets your gene of interest or the gene that you want to knock out or potentially overexpress, but I want to knock it out. And you go through all of this, you synthesize your oligos, you put them into these single guide RNA clones, you put them all together into one multiplex vector. You take all of these and put them all together into one tDNA vector that you then put into agrobacterium through floral dip. And then the importance of that extra early flowering or the flowering locus T gene becomes apparent because your successfully transformed plants will have extra early flowering because you're recruiting like so many of these activators to the site that it makes translation go really fast, and then you have lots of extra early flowering plants, and those are the plants that you know are transformed with your Cas9 construct, hopefully all of it. And then you can extract DNA and figure out which one of these plants are actually edited, because this means that the ATFT was in there and that it was working, but it doesn't necessarily mean that they have the edits in them. So then you take these plants, you sequence them, or you at least genotype them, and then you move on to the T2 generation. So you take the seeds, you plant them. In the T2 generation, you want to select for plants that have standard flowering, because that means that your Cas9 construct is no longer in the plant, but that it is edited. So in this plant, there would be no raptor, but also no Cas9, so it wouldn't keep editing. I think I'm explaining that. And then you screen them again. And this is what it looks like. So these are the extra early flowering plants. And these are the ones that you want to save and harvest seeds from to plant them in the next generation. So once I generate this raptor heterozygote or homozygote, so if our hypothesis is kind of that raptor 1b is embryo lethal. So that would mean that I wouldn't even get a plant that was this big ever if it was truly a homozygous knockout. So the plan is first of all to see if these plants are dead. If they're not, that's a whole other thing. If the homozygotes are dead, take the heterozygotes, self them, which means to just let them breed with themselves, and then you harvest seeds and pool them based on phenotype. So remember that picture from the very beginning that had the really tiny seed? That is probably what it will look like when we look at the seeds if it's embryo lethal. So we'll get basically a 3-to-1 ratio because of Mendelian genetics of seeds that would be viable and seeds that would not be viable. And that is basically one of the ways that we're going to see how lethal this is, if it is, and so test that whole question. So where is this at? This is the final plasmid. So this is, where is it? Oh no. Keep going. Nope. Back. This is this plasmid that is transformed into agrobacterium and ready for floral dipping. So this will be the next step in hopefully 2 weeks or something. What does that say? 2 weeks? 3 weeks. Oh, so this isn't the final plasmid. That's why it was weird. So this is the plasmid before the final plasmid. And as you can see, we have our spectromycin resistance. This is the Raptor 1b guide RNA. So right, left. This is our ATFT guide RNA. And this is, so these are left and right ones of these two, I think. I think there's just 2 of them actually. And then that whole scaffold bit is right here. So this is the final plasmid. It's like 26,000 base pairs big. And this is where our guide RNAs are. And that's where our Cas9 is. And it's got lots of other stuff. And this will all go into into agrobacterium, and then it will go into Arabidopsis. And then we will know what that looks like. So moving on to step 2 of determining whether or not raptor is lethal and or what its effects are when it's gone. Virus-induced gene silencing is a pretty common method for transiently or basically for silencing things that are known to be lethal or could potentially be lethal because you're silencing the gene after the plant is already alive. And so, it's not just if something is dead or not, you can actually see a phenotype if the gene is lethal or not, or what the effects are. So with virus-induced gene silencing, we use tobacco rattle virus, which is a plant virus that especially likes to be in Nicotiana. And we remove all of the infection stuff, so it's not actually causing disease in the plant. But we leave in the RNA-dependent RNA polymerase, the coat proteins, and then a spot in which we can place a 300 to 500 base pair part of our gene of interest, which in this case is either Raptor 1b or Raptor 1a. And what this does, we have our TRV, it enters the plant. The viral single-stranded RNAs are transcribed, translated, transcribed by the RNA-dependent RNA polymerase. Dicer finds them, it cuts them into silencing RNAs, and then when the plant makes its endogenous transcripts, those also get silenced because the plant is like, oh no, this is viral RNA. but it's actually not because we're sneaky and we put plant RNA in there. And now the plant thinks that that's a virus too, and it gets silenced. So that's another way to see what happens when we silence Victor. These are the current target sites for my FIGS experiments. As you can see, you sort of want to design them all over because you don't really know which one will have which effect. Essentially, these should all knock out Raptor, but, well, also these are different. So these is like 1B, 1A, and then this weird third one that we found in Nicotiana, because the genome for Nicotiana bentamiana isn't actually like great right now. And as people are getting better at it, we're starting to like find some things where maybe there's bits of Raptor, like maybe this is like almost the same, but it's not. Anyway, so these are all the big silencing targets. And what will happen is that when we infiltrate with our constructs, so you clone this into Agrobacterium, you infiltrate it into the plant. This is kind of, these are a little old, but if the gene of interest isn't lethal or if it's not deleterious, like this is what we use for our positive control, this is Gus, plants don't have Gus, it's a green fluorescent pigment. The plant will grow just fine with little defect. If the gene is lethal or if it has a deleterious effect, the plant will usually stay about this size or it will just stop growing entirely. Is that the last one? So I don't have any pictures of plants to show because they are growing. And these are all cloned into Agrobacterium as well and are waiting to be infiltrated into the plants. All right. Aim or idea 3, strategy 3 for determining the importance of Raptor in plants is to look at Salk and sail lines. So Salk and sail lines are generated through massive random tDNA insertional mutagenesis. tDNA is meant to cause a loss of function in the inserted gene. This is the Raptor gene. As you can see, these are where all the different tDNAs are inserted. And if you're wondering, this is what they used in those previous papers, is these tDNA lines. There's like a bunch of them for Raptor. These are some of the most popular. Sail lines are pretty much the same, but they usually contain multiple tDNA insertions, whereas Salk lines usually only have one. So basically, they're the same thing. They're just different ways of doing it. So why do we care about genotyping them, and how is this going to help us figure out about Raptor's importance? So a lot about tDNA insertions isn't 100% known, and sometimes it is known that tDNA insertions can cause translocations or chromosome rearrangements. They can also introduce new promoters, terminators, and or splice sites. Like in this picture here, people have done studies on this, although they're kind of limited. This tDNA array induced this whole chromosome rearrangement right here, translocation. So I guess there's more pictures too of this kind of thing. Oh, this picture is bad. Oh, let's get that. That's very blurry. So you can see tDNA integration induced large-scale rearrangements in this SAIL 232, which is a SAIL line genome, did lots of weird stuff. So one of the reasons we like to genotype them is to know whether or not any of this stuff has happened and sort of what effect it has had, because if a line is doing something weird, we want to make sure that it's actually because of the gene of interest and not because of weird tDNA things. So how do we do this? We designed 3 primers. BP, which is a border primer, targets the tDNA sequence, which is right here. LP targets the left side of the genomic sequence to the left of the tDNA insertion site, and then the genomic sequence to the right side of the tDNA insertion site with our RP primer. Then we do a bunch of PCRs, and if we get a band from these two, primers, which is LP and RP, we'll know that we have at least one copy of the wild type allele. If we get two bands, or we get one band in each spot, we know that we have a heterozygote because we have one wild type band and one mutant tDNA band. So you'll get something like that. And then if we only get a band from this border primer and this right primer, then we'll know that we have a homozygous mutant or homozygous tDNA knockout line. That's kind of what that looks like over there. So this is an example of what one of those gels can kind of look like. So this is one of the tDNA lines that, not in the frame, that I showed earlier in that little graph with the things in it. And on the left, we have our left primer and right primer. So this would be the band that we get if we have a wild type allele in our plant. And this would be the band that we get if we have a tDNA line, allele in our plant. So in this picture, just on the top here, there are two heterozygous plants and one homozygous knockout plant, which is this one, because we have no wild type band, but we do have a tDNA band. The rest are wild type. So this line, I only actually, yeah, I think, I don't know what happened here. Anyway, I did 5 plants. They were all wild type in this line. So what is the next step with this? So now we know that we have a couple of plants up here that are heterozygous and one that's maybe homozygous for the knockout for, this is Raptor 1B. This one is Tor knockout. So the seeds of these plants were harvested and they are currently plated on 1/2 MS plates with 1% sucrose and GA. So the seeds of these plants, of just the heterozygous ones, were collected. This is the T2 generation, not the G2, not generation 2. And they should segregate in this 1 to 2 to 1 ratio, but it's like, I won't know which ones of these are each one. So it's basically a 3 to 1 ratio with 1/4 of them not growing, if this is lethal. The homozygous ones should just not grow. But basically, this is the ratio that I'm trying to figure out. If this is correct, and these are actually dead, and then, well, like these will be genotype too, but yeah. So that's the fun and important part of genotyping these Salk and Say lines. This is also to shed some light on some of the results from the previous papers and a lot of the papers that make claims about Raptor, which is to say that, I mean, hopefully they're doing genotyping, but also a lot of times these seeds are not actually knockouts of the genes, even though they say they are. All right. This is my responsible conduct of research slide. I decided to do mine on data management practices. So one of the things that I think our lab does really well is how we record our data and how we share it with each other and keep it for ourselves. We record our data in multiple places. One of them is our physical lab notebooks. I'm personally a fan of this because that's what I've always used. But I have in like the last year started using an online lab notebook so that I can have like a whole sheet that other people can see. A lot of these, so one of the things that they talked about about this stuff is that how do you know that the dates are correct? So a lot of times we include pictures that are dated just to make sure that we're keeping track of the timelines of things. Attention to detail is something that's really important. Each experiment is recorded with as much detail as possible for future researchers and for ourselves, because I know that I don't remember things ever. This includes volume of reagents, type of reagents, temperatures, PCR cycles, incubation times, changes to any protocol. A lot of our protocols are shared in the lab, so whenever someone makes a change, we record that in multiple places. Light intensity, growth time, etc. All of this stuff is recorded in multiple places and in detail. Lab notebooks are stored in the lab, which is locked overnight. And then a lot of us have our own external hard drives that we back up our data to, which are also kept in the lab and locked overnight. And that is my last slide. Thank you to the NSF. I do have the NSF GRFP fellowship, like Jake said, the NIH, and the USDA. And then this is the Brunkard Lab, although Michael is now gone and Allie is gone, but that's almost everyone. Okay, I think we have time for like a couple questions, but then, yeah. But then Ryan will go.
I was intrigued by the data you showed at the very beginning of the sort of changing in phenotype over time of this Raptor 1B mutant. So it brought to mind the possibility of some modifier of that mutation. If there's some gene that, when also mutated, allows that Raptor 1B to grow bigger. So do you think that's a possibility? And is there a way to identify that modifier gene? Because that might tell you about the function of Raptor 1B.
Well, one of the prime culprits is Raptor 1A actually. So, but yeah, I actually think that would be interesting to track, is like using the same line, how as it's passed through generations, how mutations are collected and maintained in that sort of line. But I do think that it's possible that something like that has happened. That's why we try and genotype them and make sure that we know what's in there. Yeah.
So has anybody sequenced the Raptor 1A gene in those seeds that now grow bigger to see whether there's maybe a mutation in a promoter or an enhancer element that allows higher, or just look at expression of Raptor 1A?
I don't know, but that's.
You will.
I will. No, that's a good idea. Yeah. Yeah. Yeah.
How do you do
So this question kind of complements David's question. What is the similarity in sequence between the two homologs A and B?
They are similar, but less than you would think. So they're I wish I remembered now. One of them, so when you look at the expression levels of it, it almost seems over the years, it seems to be being lost, evolutionarily. It's losing parts of the gene, parts of the sequences are the same, but it's missing parts.